Hello viewers, in my previous lecture on lipids, I had discussed simple lipids, that is waxes. And we know waxes exist in nature as covering of leaves and stems. It also occurs in the brain of sperm whale. It is present in honeycombs. It is present in our ears, where it is present as ear wax. It is also present as paraffin wax. So in my previous lectures, I have discussed with you simple lipids, in which I discussed triglycerides, that is oils and fats, and waxes. Continuing the thread from there, I would now like to discuss complex or compound lipids with you. So like simple lipids, compound lipids are also esters of fatty acids and alcohols. But along with fatty acids and alcohols, there are some other groups also present in complex lipids. And the additional groups which are present in compound lipids or complex lipids may be phosphoric acid, nitrogenous base, carbohydrate moieties, proteins, or sulfate groups. So when we look into the structure, so what do we find? That on the left-hand side of the screen, what you can see is a simple lipid. That is a simple lipid. That is, here I have taken an example of an oil, example of a fat or an oil, depending on the nature of the R group here. If the R group is saturated, it will be fat. If the R group is unsaturated, it will be an example of an oil. So what we find that this is, when we are talking of a simple lipid, it is a triglyceride. That is when we are talking of an oil or a fat. But when we go on to compound lipids, what we find that one of the acyl groups of the simple lipid is replaced by a phosphate group, and this phosphate group is then joined to a X group here, to another group here, which may be nitrogenous or it may be non-nitrogenous. So this is the difference between a simple lipid and a complex lipid. So now this is the general difference. Now what we find that complex lipids can be further classified into 3 subgroups depending upon the additional group which is present there. If phosphate is present, we call them as phospholipids. If a carbohydrate moiety is present, we call them as glycolipids. And if proteins, that is amino acids, are present, we call them as lipoproteins. So let us today start our discussion with phospholipids. So what are phospholipids? Phospholipids will contain a phosphoric acid and a nitrogenous base in addition to a fatty acid and glycerol. So what do we find here? If you look at your screens, you can look at the structure of a phospholipid, and what you find here is that we have 2 acyl groups here. This may be a saturated fatty acid, and this is generally a unsaturated fatty acid. That is, the ester linkage here is between one of the oxygens of the glycerol, that is on the first carbon, and a saturated fatty acid, and the second ester linkage is between the second carbon, oxygen of the second carbon, and a ester and a carbox fatty acid, whose hydrocarbon part is unsaturated. And the third bond, the ester linkage, there is a phosphoester linkage between the third oxygen, or on the oxygen on the third carbon, and the phosphate group. And then this phosphate group again makes another phosphoester bond with this X here. And this X may be a nitrogenous base or it may be a non-nitrogenous moiety. So this is the general structure. of the phospholipid. Now when we look into the chemical structure of the phospholipid, what we find here is that this is the phosphate group here. Phosphate group is forming a phosphoester bond with the third carbon of the glycerol, and on the second side it is again forming a phosphoester bond with a nitrogenous base here, and the two groups of the hydroxyl groups of glycerol are involved in acyl linkage between the two fatty acid molecules. So when we look at this molecule very carefully, what we find that the phosphate part of the molecule forms the head. That is, and it is polar in nature because of the presence of the phosphate group here, because of the presence of the nitrogenous base here, because of the presence of the backbone here, that is the glycerol backbone here. This part is polar and it is hydrophilic in nature. Whereas when we look at the long chain of the fatty acid, we find that it is made up of hydrocarbons, and we know that hydrocarbons are non-polar in nature. So that means now phospholipid is amphipathic in nature, that is the polar part is hydrophilic, which is made up of the phosphate head, and the hydrocarbon part, which is the tail part of the phospholipid, this is hydrophobic in nature. So as a result of which phospholipid has both the parts, the polar part and the non-polar part. Therefore we say phospholipids are amphipathic in nature. So now if we look into the chemical constitution of phospholipids, we find that in phospholipids the alcohol may be glycerol or it may be sphingol or sphingosine, and the fatty acids may be PUFA or SFAs. When we say PUFA, that means they are polyunsaturated fatty acids, or they may have saturated fatty acids. So along with glycerol or some other alcohol and fatty acids, in phospholipids the additional groups present are phosphoric acid, and along with phosphoric acid we can have nitrogenous moiety or a non-nitrogenous moiety. So now when we talk of phospholipids, so what are we going to look for if they give us a question as to which of the following are phospholipids? So what are you going to look for phospholipids? You are going to look for that the molecule should be having two fatty acids, that is it should have two acyl linkages. Along with that it should have a phosphoester bond in it, that is phosphate should be present in it. And we have an alcohol part. Now this is the general thing that we are going to look when we are looking for a phospholipid. But now these phospholipids can also be further classified on the basis of the alcohol present in them. If the alcohol present in phospholipids is glycerol, we call those phospholipids as glycerophospholipids. And if the alcohol is sphingosine, also known as sphingol, then the phospholipids are known as sphingospholipids. So what you have to remember is, what are phospholipids? Phospholipids are complex lipids which are made up of an alcohol part, a fatty acid and a phosphate part. And where do they differ? They can differ in the alcohol may be a glycerol or it may be sphingosine, and then depending on the constituent as to what is the alcohol, they are known as glycerophospholipids or they are known as sphingospholipids. If the alcohol is glycerol, they are known as glycerophospholipids, and if the alcohol is sphingosine, they are known as sphingospholipids. In today's lecture, I shall be discussing glycerophospholipids in great detail. So as I've said already that in glycerophospholipids, alcohol is glycerol. And we know glycerol is a trihydric alcohol. That is, it is a triol. So what we find that two hydroxyl groups of glycerol make an ester linkage with fatty acids. Here you can see, and the third hydroxyl group makes a phosphoester linkage with phosphate group, which in turn makes a linkage with another compound X, phosphoester linkage, which may phosphoester bond here, which may be a amino alcohol or some other non-nitrogenous moiety. So what we find that two fatty acids from ester bonds with the first and second hydroxyl group of the glycerol. And the third oxygen on glycerol is bonded to a phosphoric acid through a phosphate ester bond. So also what we find, there is usually a complex amino alcohol attached to a phosphate through a second phosphate ester bond. And these complex nitrogenous bases, amino alcohols, may be choline, ethanolamine, and an amino acid serine. So this is what is the general structure. And what we find that the properties of phospholipids are characterized by the properties of the fatty acid chain and the phosphate and the amino alcohol which is present there. That means the properties will be defined by the constituents which make a phospholipid. That is fatty acid, alcohol, the phosphate group, and the amino alcohol which we are taking as another moiety. And this will make the molecule polar, this part. And we know that of course the long chain hydrocarbon chains of the fatty acids are non-polar. So this part of the molecule will be non-polar. So when we look into the phosphate group as to what makes the head polar, what we find that phosphate group has a negatively charged oxygen and a positively charged nitrogen, and this group thus becomes ionic in nature. And along with this, that is the phosphate group having a negative charge and nitrogen have a positive charge, we find that other oxygens of the ester groups are also electronegative. So these as a whole make the end, that is the head part of the molecule, strongly ionic and polar. So as you can see on your screens here, this is the head of the molecule of the phospholipid, and this is polar because in phosphate we have the phosphate group which is having a negative charge, and then along with that we have a nitrogenous part attached to this phosphate group through phosphate ester bond which has a positive charge on nitrogen. Not only this, we have ester linkages here which are again polar. So what we find that this head is polar in nature and it is hydrophilic. Whereas when we look at the long chain, that is the hydrocarbon chain of the fatty acid, we find that this is non-polar. And these are the diglycerides. That is, why are they the diglycerides? Because here the two hydroxyl groups of glycerol are attached or making ester with two fatty acids, and these fatty acids may be same or they may be different. So therefore we call this as a diglyceride here. So this part is the polar part and this part is the non-polar part. And we'll find that this structure of the phospholipid gives it some very unique properties. So this was the general structure of a phospholipid. So when we talk, now this phospholipid can be further divided into 7 types. And this will depend whether they are having nitrogen in them, or they are nitrogen moiety in them, or they are not having nitrogen moiety in them. That is depending whether they have a nitrogenous or a nitrogenous moiety. They can be classified or they can be divided into 7 types. The simplest of them is phosphatidic acid. Now, and then as we continue the lecture, we'll find that all other phospholipids are derived from phosphatidic acid, that this is the basic structure of a phospholipid. Then we have phosphatidylcholine, which is also known as lecithin, phosphatidyl ethanolamine, which is an example of cephalin. Then we have phosphatidyl serine, which is another example of cephalin. We have phosphatidyl ethanolamine, which is a plasmogen. So all these 5 phospholipids have nitrogenous bases in them. Whereas when we talk of the 6th, which is phosphatidyl inositol, which is lipositol, this does not have a nitrogenous base in it. And the third one is last one is diphosphatidylglycerol, that is cardiolipin. This is little different from all the phospholipids that we've discussed so far. First of all, it is a non-nitrogenous structure. It will have glycerol, and then this glycerol will attach to diphosphatidyle units when we'll get cardiolipin. So let us now discuss each of these in detail. So as I said, that phosphatidic acid is the simplest example of glycerophospholipid. And when we look into the structure, what is the simplest structure that we find? That we have a glycerol molecule here, and the first hydroxyl group of the glycerol is attached to the fatty acid. Second hydroxyl group is also attached to the fatty acid, and the third hydroxyl group is attached to phosphate through phosphate ester bond. So here we have an ester bond, here we have an ester bond, and here we have a phosphate ester bond. So when we are talking of phosphatidic acid, we are talking of this unit. This unit will be present in all the phospholipids, that is glycerophospholipids. So phosphatidic acid is the simplest glycerophospholipid. In it, as I've already shown you, that the phosphatidic acid has glycerol esterified with 2 fatty acids at C1 and C2, and C3 is esterified with phosphoric acid. So as you can see on your screens here, this is the glycerol backbone of phosphatidate. This is it. And then here we have one ester bond, and here we have the second ester bond. So this is diacyl, and then at the third position, here we have a phosphoester bond, where the third hydroxyl group of glycerol is attached to phosphoric acid. And what do we have now? This molecule is known as phosphatidase, or it is known as diacylglycerol, because it is having 2 acyl groups here and 3 phosphate, since the phosphate group is present at the third carbon atom. And this is the simplest unit which will be present in all phospholipids. So we say phosphatidic acid serves as a precursor for biosynthesis of other glycerophospholipids. And how is this done? This is done either by linking of this phosphatidic acid to a nitrogenous or to a non-nitrogenous base. So this is here again I have shown you the structure of phosphatidic acid, and when we find the structure of a phosphatidic acid, what you find that this is the phosphate, this is the polar head made up of the phosphate and the acyl groups, that is the ester linkages here, and the non-polar part is made up of the hydrocarbon chain of the 2 fatty acids. So when we look into the cell membranes, which is a bilayer, you will find that this is the basic unit there. That is, this is the head and this is the tail. So this comes outside, which makes it polar, that is, it makes it soluble in water, it is amphipathic, and it is hydrophilic, and this non-polar tail is hydrophobic in nature. So the first derivative that now we have to talk of is phosphatidylcholine. Phosphatidylcholine is also known as lecithin, and this is the most common and abundant glycerophospholipid in our body. So phosphatidylcholine is commonly known as lecithin. Why is it called as lecithin? Because it is derived from the word lecithios, and lecithios meaning egg yolk. So this is known as, it was isolated from egg yolk, so it is known as lecithin. The phosphatilic acid in lecithin is linked to a nitrogenous base choline to form phosphatidylcholine. So now what we have now, when we look into the structure of the lecithin, that is what we find, that lecithin contains the ammonium salt of choline joined to the phosphate by phosphate ester bond. So what we have to remember is that the basic part of lecithin is made up of phosphatilic acid, and along with that it is linked to the ester. It is linked to the ammonium salt of the choline, that is the nitrogenous part here is the ammonium part of choline. So this is what you can again see on your screens, that here we have a glycerol backbone of the choline and here what we have, this is and then here we have the phosphate group. Here you can see the phosphate group, which is attached to the ammonium salt of choline. And in this side now we have the tail, which is made up of long chains of the fatty acids, which are non-polar in nature. So what we say, that glycerol backbone along with the phosphate head makes the molecule polar, whereas this part makes the molecule non-polar. So we say that this phospholipid is amphipathic in nature. So in this case, what we find, that nitrogen has a positive charge just as in the ammonium ion. In choline, the nitrogen has positive charge and has 3 methyl and 1 hydroxyethyl group attached to it. So when you look at the structure of the choline, what do we find? That in this case, here we'll have a phosphoester bond which it is making with phosphate, and what we find in this case, since the nitrogen is attached to 3 methyl groups and to a ethyl amino group here, to a ethanol group here, we find that it has a positive charge on it. So this is known as ethanol amine also. So the IUPAC name of this compound is 2-hydroxy NNN trimethyl ethane 1 aminium. That is, we start numbering from here 1, 2. So it is 2-hydroxy and 3 of the methyl groups are attached to nitrogen. So we call it as trimethyl and 1 aminium. That is, this is where we have a positive charge on it. So where do we find this lecithin? So when we talk of lecithin, now we know that lecithin is made up of phosphatidic unit along with choline, and it is a major component of lipid bilayers of cell membranes. Not only this, it also helps in the formation of lipoproteins, it helps in the esterification of cholesterol. Choline is largely stored as PC, that is lecithin, and it is, when I say it is a neurotransmitter, how does it act as a neurotransmitter? It acts as a neurotransmitter in form of acetylcholine. So what do we find? That where is choline, that is hydroxyamine, stored largely in our body? It is stored as lecithin. And now we know that lecithin can be extracted from soya beans, and it is used as an emulsifying agent in foods. Why is it used as an emulsifier or an emulsifying agent? Because it has both polar and non-polar properties. Because of the polar head and non-polar chains, which enable it to cause the mixing of other fats and oils with water components. That is, head is water soluble and tails are water insoluble. So they can bring both the constituents together. Another very important example of lecithin is dipalmitoyl lecithin. When I talk of dipalmitoyl lecithin, what we find here is that the two acyl groups are palmitoyl fatty acids. That is, the two ester linkages are here between the two hydroxyl groups of glycerol and two palmitic acids. And what is the function of this dipalmitoyl lecithin? It functions as a lung surfactant. What do we mean by it acts as a lung surfactant? It reduces the surface tension and prevents the collapse of lung alveoli. So that means the lungs will stay flatulated, or they will stay puffed. They will not collapse, and we will not have any lung diseases or lung collapse, because this dipalmitoyl lecithin is acting as a surfactant here. The third kind of the phospholipids that we can talk of, or that we are going to discuss, are cephalins. What are cephalins? In them, the difference between a cephalin and lecithin is the difference in the nitrogenous base. In lecithins, the nitrogenous base is choline, whereas when we talk of cephalins, in cephalins the nitrogenous base is ethanolamine or serine, which is attached to a phosphate group through phosphate ester bonds. So when it is the phosphate when the unit, the nitrogenous base is ethanolamine, it is known that cephalin is known as phosphatidyl ethanolamine. And when the base is serine, it is known as phosphatidyl serine. And both of these are examples of cephalins. And what we find, that a large variety of fatty acids make up the rest of the molecule. So that means fatty acids may be different, but when we are talking of cephalins there, the nitrogenous bases will be either ethanolamine or it will be a serine, right? And where are they found? They are found in most cell membranes, particularly in brain tissues. And they are also important in blood clotting process, as they are found in blood platelets. So till now, I have introduced you as to what are complex lipids. That is, complex lipids are made up of glycerol, fatty acids, phosphate, and a nitrogenous base or a non-nitrogenous moiety. Then we've discussed three types of complex lipids today. That is, we've discussed cephalins, we've discussed lecithins, and we've discussed the structure of the simplest phospholipid, which is phosphatidic acid, from which all the other phospholipids are derived. For this lecture, I had consulted the books by Bruce, by Wade, by Solomon, and so on. Now, in my next lecture, I shall be discussing the next cephalin with you, which we will be starting with the discussion on phosphatidyl serine, which is another example of cephalins. Thank you.